So hi everyone. Thank you for joining us today with our researcher spotlight with Dr. Rama Kommagani. He is joining us from Washington University in St. Louis. So Dr. Kommagani, I'm going to hand it over to you to introduce yourself.
Hi, thanks for having me here. I am Rama Kommagani. I'm an assistant professor in OBGYN department at Washington University School of Medicine. And I'm also faculty of Center for Reproductive Health Sciences. I've been here in almost 3 years, more than 3 years in St. Louis. I did my PhD in Ohio, basically in cancer biology. And then later I did my postdoctoral training in reproductive biology with Dr. O'Malley at Baylor College of Medicine. And later I joined WashU as a faculty to focus on the women's health aspects, in particular, the gynecological conditions such as endometriosis.
Wonderful, and thank you so much for joining us today. We're excited to hear about the work you're doing. First off, I just want to ask you, what inspired you to conduct research on endometriosis?
Well, I think two things. One being the choices I made as a grad student, and then the postdoctoral training allowed me to expose to this particular disease. And then more I read, more I studied, I was fascinated. I was in a way excited on this disease, the complex nature of disease. And then the most inspiring aspect to work on this disease is how prevalent this disease is, especially the reproductive age women across the globe. And then how much it impacts the daily life of the women with this condition. And in spite of this, it's been very underfunded, underappreciated, and understudied. I think that's what really inspires me to, do my part. We all have to chip in to, alleviate the burden, the women who are having this, debilitating disease.
Yeah, definitely. And it's exciting to hear that you're inspired by the challenge, it seems like.
Yeah, the challenges, of course.
Yeah, so I know that we funded one of your projects in 2018. So I'd really like to hear, the audience would love to hear about that project and the progress you've made with that project.
Yeah, sure. So the project was studying a specific gene, which is mutated in much of the women with endometriosis. The gene is known as GREB1. And there are some single nucleotide mutations of this gene, but not much known how these gene changes can impact the disease progression. So the goal was to see how these change in the gene structure could impact the disease. So we studied that, and we made some progress to show that, in fact, the mutated form of this GREB1 gene is detrimental for the cells to grow. Especially, we tested on the cells from the lesions from the women, and then we in fact show that these mutations promote the growth of these cells. And also we did some studies with the animal models to confirm that this particular gene GREB1 is very critical for the progression of endometriosis disease. It promotes the disease progression. So I think the funding we received from the EFA in a way allowed us to do some preliminary analysis, which gave us a complete picture toward going a big grant. So I think the fund really helped us to address a small questions, but to tackle a big question.
Yeah, that's great to hear. I mean, it sounds like the EndoFound's funding is really important for that initial stage of the project, which can become bigger in the future.
Yes, that's absolutely true. And then we are now hoping to submit a NIH grant based off the preliminary results we got through the EFA funding.
Wonderful. That's so great to hear. And what do you, I know that there's more coming to this, but what would you say the impact of this research would be on women with endometriosis and to the scientific community?
We know that endometrial disease is primarily driven by estrogen. And the gene we are working is a mediator of this estrogen steroid hormone functions. So, and we know that we cannot just use anti-estrogens, systemically to combat this disease. So maybe understanding some of the proteins which mediate the estrogen action probably will allow us to go toward a more of a precision-based studies.
We're looking at potentially like personalized medicine for endometriosis.
Endometriosis, yeah, personalized, yes.
Right. Wow, that would be huge.
Yeah, because not all the women have this mutations of this gene.
Right.
But all the women who have this mutation might have the same level of disease burden. So that's what we have to study, and I think that will really help to understand how mutations in many of the genes, how they impact the overall burden of the endometriosis disease.
Yeah, and I also think, I find myself thinking while listening to you, endometriosis is it's a heterogeneous disease. It's very different in different women. So I think your research goes to show how, it is really like that.
That's 100% true. I think that's what we are heading to tease apart how these mutations might impact different kinds of endometriosis, ovarian endometriosis, peritoneal endometriosis, and how is there a difference in this single mutation to the different kind of endometriosis. So I think that's a very important question to actually ask, and I think we have to do quite a bit, and we just started.
Yeah. Great. Well, it's exciting to hear. But I know that you have other research that you've been working on, which is also very exciting. So maybe you could tell us a bit more about that work too.
Yeah, sure. I mean, as a lab which focus on endometriosis disease, we have multiple projects, and we want to tackle the disease from different aspects. So another aspect we focus in our lab is microbiome and how it impacts the endometriosis disease progression. Specifically, we are focused on how gut bacteria, which constitute up to 60 to 70% of the bacteria in human body, how it could impact the endometriosis disease. So we made progress. We published last year that antibiotics which inhibit the specific gut bacteria can stop the disease progression in the animal models. And specifically, we showed that metronidazole, a single antibiotic, can inhibit the lesion progression in the animal models. And we haven't tested them in the humanized endometriosis mouse models, but that's where we are heading. So this is fascinating in terms of the disease burden, because we know that environmental factors, genetic factors do contribute to the disease progression. But we are also adding one more dimension, that perhaps the bad bacteria which are impacted due to the diet which we consume could impact the endometriosis disease progression. Especially the lesions, they grow in the peritoneum. So the some of the factors which are released from the gut bacteria could in fact go to this lesion, and then they could promote, or they could even inhibit the disease progression. In fact, we recently found a specific metabolite which could actually protect against endometriosis. We are currently in process of publishing this work. Hopefully, very soon, the scientific we could show our findings to the scientific community. So that's pretty exciting from our part.
Yeah, and it's truly fascinating. And what do you feel the implications, I guess, let me put it this way, the long-term implications would be for endometriosis patients of these findings?
So one of the aspects we found was that women with endometriosis have changes in certain gut microbiota. So especially in the stool samples or even endometrial tissue. So perhaps we could come up with a specific panel of bacteria or the derived products from bacteria to see whether we can use a non-invasive diagnostic tool. I think that's very important because now clinically we have to do laparoscopy to confirm the disease. However, using any biological sample such as serum or stool or endometrial biopsy will allow us to at least give a preliminary indication that the women have endometriosis. Then they can go to the more invasive procedure. So that's one aspect. The other aspect is the metabolites we found, which are derived from the bacteria, can be used as a therapeutic molecule because the molecule we identified inhibits the growth of the endometriotic these lesions in the mice, but also we showed that even in humanized models, this particular metabolite inhibits the endometriosis disease progression.
Mm-hmm. Mm-hmm.
So we fairly have a good understanding even how this metabolite, inhibits the endometriosis disease progression. So I think hopefully in near future we could utilize the findings we have recently have in hand, and maybe come up with a therapeutic target or maybe a simple diagnostic tool.
Yeah, and I also just want to ask you, what excites you about this research personally?
I mean, I know many of my friends, their spouses have these issues, and I do talk to them. The most challenging aspect is how complex this disease is. Mm-hmm. While some women have more lesions, they don't have that much pain, but some women have one lesion, they have a lot of pain. So I think, and it's a complex disease. This is just not one disease. It's like ovarian endometriosis are so different from the other lesions in other parts of the peritoneum. So it's a very challenging disease even to study in an animal mouse model because different methods or different approaches have different consequences, and so it's very complex. I think that's what really drives me to work on this particular disease.
Yeah, yeah. And with that, you also answered my other question, which was, what do you find challenging? So there are many challenges there, I think, in doing research on endo.
Yeah. So one of the main challenges is the complex nature of disease, and then the not having a precise animal model which could kind of mimic the overall disease. So you have to do multiple models to understand. The other aspect is how underfunded this endo research is. I mean, if you compare to a ovarian cancer or breast cancer, probably we are underfunded maybe 1,000-fold. So it's not easy to have funds to do a complex study. You need complex experiments to conduct, and then you have to have enough funds to do this research in a deeper way, so we could understand the disease better, and that's what in general leads to some therapeutic molecules. So I think that's that I think is the one of the challenging issue. And also I think the clinical samples, obtaining the clinical samples is also challenging because of the complex nature of the disease, because you could have 20 or 30, but not all of them are same disease, and they have different pain levels and everything. It's a little bit difficult in terms of obtaining enough number of clinical samples to conduct a specific question on this disease.
So what do you feel should be done to overcome some of these challenges?
I mean, one of the aspect the EFA is doing, the Endometriosis Foundation is trying to advocate for bringing more people, more researchers together to enable the research to go forward, which I'm really thankful that Endometriosis Foundation is doing that very well, and I'm thankful that I'm part of the this group of people who really wants to change how we perceive on this disease. And I think probably we need much more collaborations, beyond certain groups or certain geographical locations. I think we need to look at this disease from a overall, like worldwide. And then probably we need more multi-center, multi-institute collaboration so that we could have enough samples and we could, exchange the ideas. I think those are the two challenging, aspects I would think right now.
Yeah, great. It's nice to hear your thoughts about that. And we're glad for you to be part of our community too. So, and what future research do you think, especially in your field, needs to be done in endo?
I think we have to do research focusing on specific type of endometriosis. I think that's very important. I think we should not cross compare the findings to one type of endometriosis to other. We have to dissect the individual disease types. I think that's very important. And I think we still have to keep on progressing towards the factors such as the genetic factors or the environmental factors. And now the diet associated factors such as microbiome bacteria, how they could drive the endometriosis disease progression. And then the critical aspect is how the genetic factors or environmental or even the diet, the microbiome, how they talk to each other in either promoting this disease or either stopping this disease. I think those are the future aspects we really have to focus on.
Yeah, so we have a lot to do.
a lot to do.
A lot, yeah. Okay, great. Well, those are all my questions. Do you have any final thoughts or comments that you'd like to share?
Yes, thanks for having me for this, nice interaction. And then I'm very thankful for the Endometriosis Foundation of America. Thank you for the support and then, the efforts. And hopefully, as a community, we should at least, do small progress and hopefully can help women who are facing so many challenges because of this disease.
Yeah, and thank you for joining us today and sharing your work. And thank you for being dedicated to endometriosis.
Thank you. Thanks for having me here.
Thanks. Okay, stop the recording.